Hi, I'm Kristen Stall, and we are going to try and see some DNA cells. So I'm going to make a lysis buffer solution, and then a protease solution, and then we're going to get some cells. Okay, so the first thing I'm going to do is I'm going to make my lysis solution. So I'm going to take 1.5 milliliters of soap. But before I do that, I'm going to go ahead and get my distilled water. So I have that in a cup labeled distilled water. So I'm going to use an extra tube to measure it out for 8.5 milliliters. So just going to suck it up until I get to the 8.5. Alright, so now I've got 8.5. I'm going to pour it into my cup that's specifically designed for this solution. Okay, and now I'm going to just suck up a little of my detergent here. So I'm going to clean out my thing. And it says that I need 1.5 milliliters. So I went ahead and I drew a line here. Okay, so we're going to go ahead and do one. Okay, so I'm going to get to the 1, and then I'm going to squeeze it out, and I'm going to give it a couple extra squeezes to make sure I get it there. Okay, and now I'm going to go to the half. Okay, and as we see, it's not quite as easy as we think to get it to the half. Okay, so there we go. And now I'm going to go ahead and squeeze, and I'm just going to suck up some of the water that's already in there to help clear out my pipette. Alright. Okay, so now I'm going to set this off to the side because I don't need that anymore. Alright, and now I need a 1/4 tablespoon of salt. So I'm going to use my quarter here. Just got this little cup so that I don't spill it all over my counter. Okay, 1/4 tablespoon, shake it off, make it even. I'm going to dump that in there. Okay. And then I'm going to use my pipette as my stirring device. Okay, I'm going to stir that until it's all mixed up and one clear solution, and hope that as it sits that the bubbles go away. Okay. Alright, so I'm going to set that one off to the side. Now I'm going to make my protease solution. Okay, so I've got another cup, labeled that. Okay, and it says that I need 10 mils of distilled water. So again, I'm going to use my measuring thing here because I think it's easier than trying to go super close to this one here. Okay, so I'm sucking up my distilled water. And adding it until I reach the 10, because that's what I'm told this one needs. Okay, and I'm at 10, so I'm going to get rid of my extra fluid, and I'm going to dump this in here. Alright. Okay. And now I need 1/8 tablespoon of food tenderizer to help with that next step. Okay, so again, I'm going to shake it off, make sure it's even. Okay, and then I'm going to dump that in there. And put the lid back on because I will tip that over. Okay, and then I'm going to use the same pipette that I used to get the 10 mils, and I'm going to go ahead and stir this up until it's a single solution. Alright. Okay, there we go. So now I'm going to set that one off to the side. Okay, now I need to go ahead and I need to get my cells so that I can get some DNA, right? Okay, so again, my directions tell me that I need 3 mils of water. Okay, so I've got some tap water here. Okay, and another pipette that I'm just going to use to fill this up to 3 mils because I'm one that counts on recipes and I was given one, so I'm going to use it. Alright, so we got 3 mils. Okay, so I'm chewing on my cheeks a little bit, trying to speed up this process just a little bit, right? So, okay, 30 seconds has passed, right? Okay, now my cells are in there, right? Okay, so I'm gonna move my tap water off to the side and I'm gonna empty out my pipette. Okay, now I have my test tube that I'm using specifically for my cells. Okay, the other one was just to measure. Okay, so now what I'm going to do is I'm going to take my cells that are in this water that I just swished around my cheeks to get all those cells that I just broke off by chewing on my thing. Okay, and I'm going to add those to my tube. Okay, and make sure that I got all of those, right? Set this off to the side. Okay, so now I've got that. And now my next directions is to add 2 mils of my lysis buffer solution. Okay. So again, that's why I drew a line on there so that I would make it would be easy for me to see. Okay, so I'm going to do 2 of these. Okay. Hard to see without the bubbles in there. Okay, there's 1. Squeeze it out. And then 2. Okay, give it a little squeeze. Alright, so now I've done that, and my directions tell me to invert it 5 times. Okay, so 1, 2, 3, 4, 5. Okay, and now what I'm going to do is I'm going to kind of take a look at it, and to me it just looks like hazy blue solution. Okay, alright, so now my next thing is to add my solution to break up the cells, right? Okay, so it says add 100 UP. Okay, so the first time I tried this I did a full mil, okay, but it's actually to this first line here, right there. So it might make it a little bit different this time around. Alright, so, and I did not draw a line on this one, so I look super close. Okay, here we go. Okay, so add that to the tube. It says invert 5 more times. 1, 2, 3, 4, 5. Okay, and now I'm supposed to let it sit for 10 minutes. Okay, so I'm just going to let it sit here for 10 minutes. Okay, so we waited 10 minutes and now we're back, and I need to look at it to see what changes I've noted. And to me, it really just looks, it's not really much blue, so the blue is lighter, and it almost looks like there's a line of blue up the top, so maybe a layer. Okay, so my next directions are to make my DNA available. Okay, so I got my alcohol out of the freezer as was suggested, and I've got a pipette that I'm using only for this alcohol so that I didn't have to pour it into a cup as well. Okay, and so it says I need to add it till I get to the 10 ml line. Okay, so I'm going to suck it up. Okay, and then I'm going to tilt it to the 45-degree angle as it suggests and let it run down the side until I get to the 10 ml line. first time I did this, I accidentally did that wrong too. I added 10 mils versus going to the 10 mil line, so it was much fuller the first time. Alright, and I'm just going to let the rest of that be in there. Right cap. Alright, okay, so now I'm supposed to take a look at it and see what I see. Okay, now what I see is kind of some floating, almost looks like little bubbles, but I'm going to guess that that's some kind of cells or maybe DNA starting to separate itself. Okay, but my instructions say to let it sit for 5 minutes, so we're going to do that and we'll be back in a second. Alright, so we went ahead and let it sit for 5 minutes as suggested, and what I see is some of those things are now floating at the top. So that's also a little different than the first time I did it. The first time they were hanging out in the middle. Okay, so we got some, I'm going to call them floaties for this at this moment, but I'm pretty sure that that's my DNA that has separated out. Okay, so it says look at where the layers meet, and it does kind of look like there is a thin layer of something in between. It's like a little, I don't know how to explain it. I'm going to have to think about that so that I can write it in my notes. So there is a little bit of layer of something, but there's definitely two distinct layers with the clear and the cloudy. Okay, so now it says to invert it 5 times. Okay, so 1, 2, 3, 4, 5. Alright, so now what do we see? I see a whole lot of floaties, and I'm going to think that those floaties are my DNA. Okay, so it looks like some of them are kind of long and stringy, and some of them kind of look like little parts, or there's like brighter parts than others. Okay, so that is my experiment, and now I see my DNA. Thank you.